Good morning, everyone. We are starting with the new topic that is the lipid metabolism. Before going on to the individual subsections of lipid metabolism, I would like to share this particular slide with you people that is about how to write a long question of any metabolism. So you can divide a long question in following subheadings. First is the definition where you have to write the name of the substrate and the final product. So for example, if you want to define glycolysis, so glycolysis is the process where glucose is getting broken down into two molecules of pyruvate. Second thing which you have to write is the site. So in site you have to write the major organs or metabolic organs which are the sites where this metabolism takes place. So mostly they will be liver, adipose tissue, kidney, skeletal muscle, etc. Subcellular site where you have to write about the part of the cell where metabolism is taking place. So whether it is cytosol or it is mitochondria or it can be lysosome, etc., etc., that you have to mention. Other name of the pathway that also you have to remember. So suppose Embedon-Mayer pathway, which is another name of glycolysis, that also you should remember. Then the major steps of the pathway where you have to write the complete reaction. So complete reaction will include substrate, product, name of enzyme, cofactor, etc. Also you have to draw the direction of the reaction, whether it is in one direction or it is reversible. Then energetics, where you have to mention the number of ATPs which are either getting produced or getting utilized, net ATP generation that you have to write separately. Regulation, regulation by substrate or hormone that you have to mention. And finally, the disorder associated with that pathway. So if a question is of 10 marks, then you have to divide the metabolic pathway among the following headings, and then you can start writing the long question. Okay, fine. Now moving on to the our main topic, that is the lipid metabolism. So first important short note in this particular chapter is digestion and absorption of lipids. So all of you know that metabolism is a three-stage process. So in three-stage process, first step is digestion and absorption, where any molecule is getting converted to in simple form. So suppose in carbohydrate metabolism, polysaccharides ultimately was getting converted to glucose and then glucose was getting absorbed. Okay. Similarly, in proteins, first protein has to be converted into amino acid and then they goes under the process of metabolism. So that is the first process. Second step of any metabolism is the main reactions or oxidation processes which gives rise to formation of acetyl CoA or acetyl coenzyme A, which is the junction point of all the three main metabolism, that is the carbohydrate, proteins and lipids. Ultimately they will produce acetyl CoA. Third step is TCA cycle, where NADH2 or FADH2 are getting produced. They goes to ATC and generates the ATP. Okay, so this is the process by which we are getting energy from our main molecules, that is the carbohydrate, proteins and lipids. So you have already learned about the carbohydrate and protein metabolism. So similarly in lipid, we'll start with the digestion and absorption of lipid, where we will convert a complex lipid molecule into its simpler form. Okay, fine. So all of you know the dietary source of lipids includes milk, milk products such as the curd, paneer, cheese, etc. Apart from this, there are certain non-vegetarian sources like fish or liver. They are the main sources of lipids in our diet. Also, the major sources of trans fatty acids in our diet includes all this junk food or fast food, which we love, but it is mainly rich in trans fats. So consumption of this type of food should be kept minimum, or one should avoid. Okay, fine. Normally, we take around 50 to 150 grams of lipid in a day. That is the mainly daily requirement of amount of fat that is needed. Out of this, 90% is triacyl glycerol, or commonly known as the TG. Remaining 10% includes other form of lipids, that are the phospholipids or fatty acids or cholesterol esters. But this TG represents the dietary lipid. Okay, and it is the main part of our lipid panel. So suppose if you give any sample for lipid panel, in that there will be several other parameters are included, such as the TG, LDL, HDL, total cholesterol, etc. Out of this, triglyceride level represents how much lipid you are taking or fat you are taking in the form of diet, and its level should be less than 150 milligrams per deciliter. Okay, more than 150 or more than 200 is harmful for our body, and if your triglyceride comes more than 200, then you should start take doing restriction in taking lipid content in your diet. So sources we already know about, from where we get the lipid or fat. Importance of lipids, as you know, that first and most important will be the energy. 1 gram of fat gives us the 9 kilocalories. Then triglyceride, which is the emergency source of energy. The triglyceride is getting stored mainly in the adipose tissues of our body, and depending on certain situations such as the starvation, where the stored triglyceride gives us the energy, it goes under the process of oxidation. Apart from this, phospholipids, which are the main form of lipids, their function is already known, that lecithin is act as a surfactant. It can act as an insulation. It helps in formation of myelin sheath. Cardiolipin, which act as an antigenic property. They also helpful in formation of biological membrane. Cholesterol, which is an imported product, which is the one kind of lipid only. It helpful in formation of steroid hormones, bile acids, as well as the vitamin A and vitamin D, fat soluble vitamins that are getting produced from the cholesterol. Apart from this, the conjugated or complex lipids also include the lipoproteins, where proteins are attached with the lipid, and it is must for the transportation of lipid across our body. So importance you have already learned under the chapter of chemistry of lipids. So this is the main form of lipid in our diet, that is the triacylglycerol. In this, there will be a ester linkage between this glycerol molecule and fatty acid. So this is the structure of TG or triacylglycerol. Since it has three acyl chain or fatty acid chain, in its structure attached with the glycerol. So that's why it is known as the triacylglycerol, or commonly known as the TG. So if there is a breakdown of this triacylglycerol or TG, obviously there will be release of two compound, glycerol and fatty acid. Okay. So in process of digestion, we have to convert this triglyceride which we take from the diet into glycerol and fatty acid. Main enzymes which are involved in this process. So process of digestion starts from the mouth, where the primary enzyme is lingual lipase, which is usually released from the dorsum of the tongue by Abdon's gland. Now, this particular enzyme has a major role in infants and in young children, where the main source of lipid is through breast milk. So breast milk will contain short and medium chain fatty acids, and digestion of this fatty acid can be taken place in the mouth by lingual lipase. Second enzyme which will be helpful is the gastric lipase. So as its name suggests, it will be present in the stomach. It will be secreted by the parietal cells of the stomach. It helpful in digestion of long chain fatty acids. Third organ which is involved in digestion is the intestine, where the main enzymes will be lipase and colipase, which are part of pancreatic juice. Apart from this, pancreatic juice will also contain phospholipase A2 and cholesterol esterase. So they will act correspondingly on phospholipids and cholesterols. Apart from this, we require help of bile salt in the micelle formation. So detail about it we will discuss as we discuss this particular topic. Okay, fine. So first is the lingual lipase, which is released by the dorsum of the tongue. It is secreted on the saliva. So in mouth, very little digestion of the lipid takes place. Mainly this particular enzyme is getting activated in the stomach, where pH is slightly on the acidic side, around 2.5 to 4. Okay, so main it's what does it do? It splits the triacylglycerol into fatty acids and glycerol. So whatever lipase which we talk, ultimately they will convert your triglyceride into its two main part, that is the glycerol and fatty acid. The thing which you have to remember is the site where these lipases are acting on triacylglycerol. Secondly is the gastric lipase. So they digest around 30% of total triacylglycerol. They act on third ester linkage of triacylglycerol, mainly of short and medium chain fatty acids. And they will produce 1,2-diacylglycerol and free fatty acid. So where they will act, this gastric lipase will act at this particular linkage, and it will release this particular fatty acid. Okay. So if this fatty acid is getting released, then the remaining product will be 1,2-diacyl glycerol, because we have acyl chain at the first and second carbon. So this particular product is known as the 1,2-DAG, diacyl glycerol. Simply you can remember the diacyl glycerol as a product of gastric lipase, and free fatty acid is another product. In intestine, as we mentioned, that pancreatic juice plays the main role in digestion of lipid. So the enzymes which it contains include pancreatic lipase and colipase. Apart from this, it contains another enzyme, phospholipase A2 and cholesterol esterase. Along with these enzymes, there will be also presence of bile salt, which are mainly produced in the liver. From liver, it can come into the pancreatic juice, and along with the action of lipase and colipase, it helpful in lipid digestion. So pancreatic lipase, they will act at the first ester linkage and the third ester linkage of triacyl glycerol. So they will liberate the fatty acid present on the first carbon and the third carbon. So remaining product will be 2-acyl glycerol. So there will be fatty acid present at the carbon number 2. After that isomerization takes place. So this fatty acid is getting shifted at the first carbon. So the product will be 1-acyl glycerol. If you can see here, here it is at the carbon number 2. Now it has been shifted to carbon number 1. After that, once again, the lipase will act and it will release the fatty acid from the first carbon, and final thing will be the glycerol molecule. Okay. So here also ultimately the product will be glycerol and fatty acid, but in case of gastric lipase, it was diacyl glycerol. Here it will be free fatty acid and glycerol. Okay. So in digestion, mainly what we talk about is that TAG was either getting converted to free fatty acid, glycerol, or 1,2-diacylglycerol or monoacyl glycerol. Okay, so these are the various products of the triglyceride which we get after the process of digestion. Another form of lipid which we take, that is the phospholipid, on which our enzyme phospholipase A2 will act. It will give us the lysophospholipid, or suppose our phospholipid is lecithin, then the product will be lysolecithin. Another thing is the cholesterol ester which we take in the diet. On that, esterase enzyme will act and it will give us the cholesterol plus free fatty acid. Okay. So other form of digestive product will include, apart from this, will be lysophospholipid and cholesterol and free fatty acid. Okay, fine. So all this digested product of lipid, they will combine and will start the next process of absorption. So here what it will do, there will be formation of a compound, formation of all this digested product and bile acids which are produced in the liver. Okay. More about these bile acids or bile salts we will discuss when we discuss separately about the metabolism of bile acids. But right now remember that these bile salts or bile acids, what are the bile salts? Basically bile salts, they are derived product of glycine and taurine. You already know what is glycine. Glycine is an amino acid. So sodium and potassium salt of bile acids, which are ultimately the product of glycine and taurine, they are known as the bile salts. The common names of the bile salts are the sodium and potassium glycolate and sodium and potassium taurocolate. Okay. So detail about this you have already learned in abnormal constituents of urine, and also we will discuss separately about the product of bile salts in detail in our upcoming lectures. Okay. So these bile salts along with this digested lipid, they will form a structure. That structure is known as the micelle. Okay, or commonly it is known as the mixed micelle because it is a mixture of so many things. So efficient lipid digestion requires micelle. So what does this bile salts actually do? So bile salts reduces the surface tension by increasing the surface area for the actions of enzymes. So here, suppose this is a large fat globule. Our bile salt will act on this, and they will convert this large fat molecule into small globules, something like this. So this fat globule ultimately will be converted something like these fat globules. So because of that, the surface area or surface or area for enzyme to work gets increased. Initially, for suppose for any lipase, the area which is available was only this. But after the formation of globule, this area is already there. In additionally, it gets the this much area to work on. So the process of digestion gets simpler and more faster. So this process of converting large fat globule into the smaller or tiny fat globules is known as the emulsification, and that is taking place in our body with the help of bile salts. Okay, so this particular question also can be asked in the practicals, when in the practicals of abnormal constituents of urine. Okay, so this emulsification is process is must for the smooth digestion of lipid products, because lipids are not digested as quickly as carbohydrate or proteins. Okay, so this is the micelle structure. You can see here, you can draw simple micelle structure like this, where you have to mention about its component, that is the phospholipid, monoglycerides, long chain fatty acid, bile salt, cholesterol, etc. Okay, so you have to mention individually about phospholipid, monoacyl glycerol or monoglyceride, long chain or free fatty acid, bile salt, and cholesterol. Okay, you can draw this kind of simple diagram of mixed micelle. So what does this micelle actually do? Micelle, which is a mixture of digested lipid product and bile salts, okay, it can act as an anchor. It helps in smoothing of action of our digestive enzymes, that is the colipase and lipase. Because of this micelles, this colipase and lipase will act on our triacylglycerol, and they will helpful in generation of free fatty acids and glycerol more quickly. And this micelle, which also contains digested product, it comes in contact with the mucosal cell, and it releases digested product into intestinal mucosa for the absorption process. Okay, so this from lumen it comes to the mucosal cells. So micelle will release all the digested lipid products, and the bile salts will once again come back into the circulation for another micelle formation. So basically, micelle act as a carrier for digested lipids to getting absorbed. So it's a intermediate process between digestion and absorption of lipid. that you have to remember. Okay, fine. This is another diagram which is showing the same thing, the component of micelles in a better way. Okay, fine. So another forms of lipids which we take in the diet apart from TG, that is the cholesterol esterase. So in that, if you see cholesterol ester, that will be a mixture of cholesterol and free fatty acid. So this fatty acid will be released by action of cholesterol esters. Phospholipids, which we take in the diet, for example, if it is lecithin, then it will be hydrolyzed into lysolecithin and free cholesterol by enzyme phospholipase. There are several varieties of phospholipase, but you don't have to go in detail. Okay, fine. So this is the table which showing the summary of digestion process, where the end product will be free fatty acid, diacylglycerol, monoacylglycerol, lysolecithin, cholesterol, etc. About these two enzymes, lipoprotein lipase and hormosensitive lipase, we will discuss separately when we talk about lipoprotein metabolism. Okay, fine. So you can understand the same by this particular diagram, or you can represent the thing in this particular way. You don't have to draw any structure. Okay, so the process of digestion start from the mouth in lingual lipase form, then in stomach there will be gastric lipase. These two mainly will act on short and medium chain fatty acids. In pancreas we have pancreatic lipase. It will act on long chain fatty acids, and our products which are free fatty acid, monoacylglycerol and cholesterol, as well as the lysophospholipids. All this will combine with the bile salts. They will form micelle, and micelle will release the product into the intestinal mucosa. Okay, fine. Now coming to the process of absorption. So our process of absorption takes place mainly in the jejunum. Okay, so digested lipid products which are packed into the micelle will move towards the intestinal mucosa. All the digested lipid will be released except bile salts. Bile salts undergo enterohepatic circulation for the reutilization process. Now remember here, the glycerol and short and medium chain fatty acids, they can be directly absorbed and transported to the liver for the reutilization. Okay, this particular thing is mainly for the long chain fatty acids. Okay, fine. In absorption, these are the steps which takes place. So in absorption, the thing which we have to remember here, whatever the products of the triacylglycerol, now once again they will be repacked in the form of triacylglycerol. Okay, so first there was an unpacking of triacylglycerol into monoacylglycerol or diacylglycerol. Okay, fine. Now once again they will be packed into triacylglycerol, because these digested products will be once again gets converted to TAG, and this TAG will be incorporated along with the proteins, that is the ApoA and ApoB48, and they will form lipoproteins. Okay, and as we know that lipids cannot transport on its own, it requires conjugation with the protein to get transported into blood to various organs. Okay, so all this triacylglycerol which has been absorbed will be transported in the form of chylomicron, and the metabolism of chylomicron we will understand separately. Okay, so this 2-acyl glycerol once again will be converted to triacyl glycerol. 1-acyl glycerol will be broken into free fatty acid and glycerol. So this long chain utilizes this glycerol and it will be converted to triacyl glycerol. The glycerol which has been absorbed will go directly to the liver for the formation of essential products. Okay, fine. So if we see here what happens, these are our digested product. With the help of micelle, they will be come into the intestinal mucosal cell. Okay, fine. So this free fatty acid and glycerol, they will combine, can form triacyl glycerol. Apart from this, this 2-acyl glycerol will bind with free fatty acid and form triacyl glycerol. Okay, so first there was unpacking of Tg, and now there will be once again packing in the form of TAG. Okay, this triacylglycerol along with the phospholipids, cholesterol, and ApoA and ApoB48, that is the protein. Okay, protein plus lipid will form lipoproteins. That lipoprotein is known as the chylomicron. Chylomicron will be taking this all these lipids in the our lactal glands, mainly the thoracic duct, and from thoracic duct it will come into the circulation and it will transport towards the different organs. Okay, fine. So that is about the long chain fatty acids. Small medium chain fatty acids can be directly come into the circulation. Okay, so in absorption part you have to remember there is a packing of triacyl glycerol once again which was unpacked. Coming to the disorder part. Okay, so the disorder includes, first is the steatorrhea. Steatorrhea is the excretion of fatty foul smelling stool. So basically what does it say? That undigested lipid can be excreted in the form of stool and they will give foul smelling of fat. Okay, that is known as the steatorrhea. So what can be the causes of steatorrhea? It can be at the digestion level or it can be at the absorption level. So suppose if we understand the causes by this particular diagram, so if there is a defect or if there is a disease of liver, such as suppose if we say hepatitis or cirrhosis or any tumor, then it will hamper the formation of bile salt because bile acids are getting produced in the liver. Okay, so low production of bile acid or bile salt also can hamper the process of digestion as well as absorption. Suppose if there is a disease of gallbladder such as the gallstone, then another thing is the cholecystitis, which is the inflammation of gallbladder. Okay, or suppose if there is a carcinoma, then also there will be hampering in the release of bile acids. Another, if there is a disease of pancreas such as the carcinoma or pancreatitis, then there will be defect in the lipase. So pancreatic lipase will not be able to work or it will not be produced in the sufficient amount. So ultimately it will prevent the digestion or it will hamper the digestion of lipids. Another thing, it can happen at the absorption level, which is at the intestine. So suppose if there is a enteritis, which is the inflammation of small intestine, if there is a infection. Another thing, if there is a autoimmune disease such as the tropical spree, okay, or Crohn's disease. In this, there will be production of autoantibodies which destructs the intestinal mucosal cells. So absorption of the dietary lipids will be hampered. So in that case also, the unabsorbed product can come into the feces, and it will give rise to steatorrhea or excretion of undigested fat into the stool. Okay, so these are the few causes of steatorrhea that you can remember. Okay, try to remember logically, try to remember all depending on the organs which are involved in the process of digestion and absorption. Okay, fine. Few other things that related with the digestion and absorption defect. One is the chyluria, that is also known as the milky urine. So sometime what happen, there will be abnormal connection between the lymphatic drainage of intestine and urinary tract. So because of that, the undigested lipids can come into the urine. Okay. Another is the chylothorax, where chyle may leak into the pleural cavity from the thoracic duct because of any injury to the thoracic duct in the form of crush injury, or if there is an error by surgical repair of any chest operation or any stab wound. Okay, all this can damage the thoracic duct, and thoracic duct may leak its chyle into the pleural cavity, and our thorax will give hallucination, hallucination appears on the X-ray. Okay. Another thing is the cholelithiasis, that is the formation of gallstone. The impact of gallstone we already discussed in the form of steatorrhea. Okay, fine. So these are the few images. If you can see here, this is the normal urine sample. This is the sample of chyluria. You can see the milkiness. This is the chylothorax, X-ray of patient of chylothorax. You can see the pleural cavity. There is so many haziness. That is because accumulation of chyle in pleural cavity. Also, on urine examination or stool examination, we can see this kind of globules. Okay, these are the fat globules which we don't see in a normal urine sample. Okay, so these are the findings or few disorders you have to remember which are associated with the digestion and absorption, and separately into marks which are, a question can be asked that write about the steatorrhea or chyluria or chylothorax. Okay, so you have to remember that also. Okay, another one thing, that is, nowadays one medicine is available which is known as the orlistat. So this orlistat is a medicine which is useful in treatment of obesity. What does it do? It inhibits the pancreatic lipase action. So if there is a low action of pancreatic lipase or no action, then fat, though it will remain undigested, but they will not be absorbed. Okay, because we absorb the digested product of the triacylglycerol. So if TG is not getting broken down, then this undigested TG can be released directly in the urine or in the, mainly in the stools, and give rise to steatorrhea. Okay, so there will be a side effect of that steatorrhea, but the fat intake, whatever fat intake we take, it will not give us the calories. Okay, so it will reduce the calorie. Of course, this kind of medicine requires several monitoring of certain levels of the fat. Also, it requires continuous monitoring of the side effect of the medicine. So more about this you will learn in pharmacology, but right now one particular medicine is available which is based on the concept of the digestion and absorption. Okay, so these are the last two slides where you can summarize the whole chapter, or you can write the whole chapter in this two flow chart only. It is there, it's taken from the Lippincott book. Okay, so you can revise the particular chapter by these two particular flow chart. Okay, fine. So that was about the digestion and absorption of lipid, which is also an important short note. Thank you.